הצילום הכי חשוב. Okay, good morning. My lecture today will be focused on nanoscale materials-based devices and the way to use those devices in biological diagnostic applications. I will start shortly with a very short background about nanotechnology. Then I will show some background about silicon nanowires and in general nanowires, how we can synthesize the nanowires and how we can apply them to build sensors, biological and chemical sensors. And I will give two examples. The first example is detection of proteins, and the second example is detection of viruses. So generally, nanotechnology is a field that is a very broad area of science that is composed of chemistry, biology, physics, engineering, and it deals mainly with the synthesis, the preparation, characterization, physical and chemical characterization of nanoscale materials when at least one of their critical dimensions is less than 100 nanometers. So it can be zero-dimension structures like nanodots or nanoparticles. It can be one-dimensional structures like the examples I will show today, nanowires. And it can be also two-dimensional monolayers when the thickness of the monolayers is less than 100 nanometers. But nanotechnology is not only deals with the preparation of nanostructures and with the characterization, but more and more it deals with the capability to use those structures, manipulate the structures, and learn how to do all those processes in order to apply them and build from them larger systems. So that's the thing I do. And also the connection between nanotechnology and biology, and that's the main area that I focus during my research. The main, the connection between biology comes. We can see this scheme and see that nanostructures like nanowires or quantum dots are very similar in their sizes to chemical and biological components. So many years ago, people thought that the interaction between chemical and biological materials and nanostructures will lead to improved sensors or sensing systems. So a sensing system is actually, we can see a very general scheme of a sensing system. It always includes a transducer, in our case a solid surface. On the transducer, we modify what we call a recognition layer. This layer is a chemical or biological layer that should be able to bind specifically chemical or biological molecules, and as a result of the binding event, we should get a signal that we can quantitate. So one of the most important things about a sensor is that the binding leads to a sensing signal. So we need a communication between the binding event, or we need actually a communication, direct communication between the sensing event and the transducer surface. So why to use actually silicon nanowires? We saw before that silicon nanowires, their size is very similar, and we will see later that it's very similar to most of the chemical and biological species that we would like to sense. We can control actually all the properties of nanowires. We can control the length, we can control the diameter, and we can control the chemical composition and electrical characteristics. We can prepare, as we will see later, arrays in very small areas of a very large number of devices. Okay. So in this scheme, we can see how we synthesize the silicon nanowires. We start from a solid surface. We deposit on this solid surface a catalyst. In this case, the catalyst for the synthesis of silicon nanowires are gold nanoparticles. We introduce this surface into a chemical vapor deposition setup. We heat the system in the presence of silane gas as a precursor. And at a certain temperature, we get the growth of silicon nanowires. So actually, we can see from this scheme that the time of the growth process can be controlled, and we can control the length of the nanowires that we get. And by controlling the size of or the diameter of the catalyst particles, we can get a controlled diameter of silicon nanowires. And we see that during the growth process, actually the gold particle gets away from the surface as the silicon nanowire grows. So we get actually a tip, a silicon tip, crystalline tip, with a gold nanodot at the end. And actually, when we remove it from the CVD setup, we get a thin layer of native oxide around the silicon structure. So the temperature of the growth is called eutectic temperature. Eutectic temperature, at this temperature, actually, the gold droplet is in the liquid form. It mixes with a Silane gas. This composes a Silane gas inside the liquid droplet, and then we get what we call unidirectional axial growth. So actually, the direction of the growth depends on the nature of the surface that we use. If we use a silicon wafer that has a silicon oxide layer, amorphous layer, then we don't get any we get a random growth of silicon nanowires in all the directions. There is no epitaxy. But if we want the silicon nanowires, we can control also that silicon nanowires grow in pre-designed directions depending on the nature and the crystallinity of the surface that we deposit the gold nanoparticles on. So we see that, again, from the synthesis, we can control the chemical composition, we can control the physical dimensions, diameter and length of the nanowires, and we can control the electronic, conductive, and optical properties by actually not only adding the Silane gas, but adding other gases, impurities that we call dopants. We can dope the nanowires to P-type nanowires, N-type nanowires. A lot of the work that we want to do today is actually make novel materials based on gradual doping during the growth process to get novel devices and novel conductive characteristics that couldn't be done today in this scale. So how people in the past prepare devices from nanowires generally? They took the nanowires from the growth surface. That's a SEM picture from a growth surface of nanowires that grows randomly on a silicon oxide surface. We need to remove the nanowires from the surface. Actually, people sonicate the nanowires, put them in a solution of ethanol or other alcohol, and then deposit them by drops on a surface that is pre-numbered. And then, by using E-beam lithography, electron beam lithography, they write electrode by electrode. They actually prepare device by device, writing electrode by electrode on nanowire by nanowire. And then we get the device that is actually a transistor, where we get two electrodes. We call them source and drain, and a single nanowire or multiple nanowires, depending on the deposition properties. We get multiple or single nanowire connecting between the two electrodes. So actually, we can see that this process is very time-consuming, and actually, all the devices that we prepare will be different one from the other. Actually, as I said before, we prepare nanotransistors or transistors. Actually, a transistor is a device where we have two electrodes. In between the two electrodes, we have a semiconductor area and a gate electrode, a third gate electrode. And by changing the potential of the gate electrode, we change the conductance between the source and the drain electrodes. For P-type devices, potentials negative than a certain value, in this case 0, cause an accumulation of carriers and an increase in the conductance of the transistor. In the case of N-type doped devices, we get the opposite behavior. The question is how we can use this kind of devices in order to create biological or chemical sensors. The thing we do is actually we create what we call a CHEMFET, where we remove the gate electrode, and we use the charging intrinsic characteristic of biological and chemical molecules to charge the surface of the semiconductor upon binding to it. And the charges, intrinsic charges of the biological or chemical molecules will act as nano gate gating electrodes. So actually, we can see here the electrical performance of a p-type device. Negative voltages cause an increase, in this case, a negative increase in the conductance of the device. But I asked the question, what we still need to create reproducible silicon nanowire devices? We need a process that is easy, the number of steps is small, and that we can get a large number of devices with very similar electrical characteristics in a very small number of steps. So what we do actually after growing the nanowires, we remove the nanowires, put them in solution, choose particular techniques to deposit the nanowires, and this is one of the, I would say that this is one of the most critical steps here, how to actually manipulate the nanoscale materials, actually locate them exactly in the areas where I want and at the density that I want, that each one of all the 200 or 400 devices in very small areas, we have at least one nanowire in between the source and the drain electrodes. So actually, I would say that this is one of the challenges today of nanotechnology, actually to take the nanoscale materials and being capable of taking them, manipulate them, and actually telling the nanomaterials, you will stay there, and not doing like most people do today, a random process where they deposit the nanowires and then they put electrodes, and most of the devices do not contain any nanowire in between the electrodes. So in this case, I use a technique that is called Langley-Blodgett. Langley-Blodgett are actually two plates, and we have two phases. We have aqueous liquid phase. The nanowires are very hydrophobic, so actually when we put them on the water phase, on water, they float on the water phase, between the air phase and the water phase. And when we compress them between the two plates, actually we align them. We compress them. We can control the density of the nanowires in between the two plates, and we actually align the nanowires perpendicularly to the force that we apply. So after we align the nanowires, by simply one photolithographic step, using a simple photomask, we can actually create in a very small area of, in this case, 6 millimeter channel. By half a millimeter, we can put more than 200 devices, electrical devices. We see that each row contains, in this case, many of those devices. We see a nanowire, and in this case, 2 devices. So in this case, we have more than 200 devices. So actually, we can see some of the experiments. What happens when the deposition, the critical, what I call the critical deposition approach doesn't work. So in many cases, when we deposit nanowires, we can see that what we call flow alignment. Nanowires can be aligned to some extent, but we cannot control the places where the nanowires are. In many cases, the nanowires aggregate when the concentrations are too high. So definitely those two cases won't lead to any satisfactory or some good device. In this case, the number of nanowires is so small on the surface that most of the devices won't have any nanowire in between the electrodes. So if I have time at the end, we are developing in our lab a new technique that we call new dry transfer techniques, where we actually do not deposit the nanowire from solutions, but we grow the nanowires at certain densities, at a certain alignment from the beginning on the growth surfaces, and then we make a printing process, and we transfer them directly inside windows that we create photolithography, and we know that just inside those windows we will have nanowires. Then we wash all the photoresist around, and we get nanowires just inside the windows. So then, by using those novel techniques, we can get that every one of all the devices will have at least one nanowire in between the electrodes. So now we see here the reproducibility of the devices. Most of the devices behave very similarly electrically. The devices act very well as field effect transistors in water. And there are many critical technical points that I don't have time to enter into them, but in order to make those devices functional in water solutions, This is really a big challenge. We need to be able to isolate all the metal components of the chip that will be stable underwater physiological solutions in order to make good sensors later on. So the first example that was developed was a pH sensor. Actually, every silicon nanowire contains silanol groups. Silanol groups are SiOH groups that can be protonated and deprotonated depending on the pH of the solution that we are working. So actually, simply by changing the pH, we change the extent of protonation and deprotonation of the surface. We change the charge of the surface of the silicon nanowire, and by doing that, we change the conductance of the field effect transistor. We see that by changing the pH, the conductance of the field effect transistor changes. Actually, a p-type device, in a p-type device, when the pH decreases, more positive charges we have on the surface, the conductance of the silicon nanowire device decreases. For an n-type device, we have the opposite behavior as we expected, as the same charges will cause an opposite change in the conductance of the device. The next step is the detection of actually proteins by using the same devices. And the big challenge here is to use what I call the multiplex detection, to be able to sense multiple proteins simultaneously in solution using a single chip. So I won't enter now into the many techniques that people use today for the detection of proteins. There is QCM that actually senses the change in a mass of a piezoelectric crystal upon the binding of any mass to the crystal. Then we have ELISA techniques that just, we get this, many biologists use still this technique. We use enzymes, other colors to get on ELISA plates. And then we have surface plasmon resonance and many other techniques that people use today. So actually the two models that I will show, the first model is the sensing of cancer markers by using silicon nanowire detectors, and the other will be bacterial toxins that actually are also proteins, but the chemistry that I had to do in order to sense those proteins is a little different. So it will actually exemplify that we can make many types of different chemistries and many types of different monolayers on those devices and how to do that. So actually this scheme shows actually the chemical modification of devices. Here we have a device, 2 electrodes, a single nanowire, silanol groups. So the first step is to bind a monolayer or a very thin layer of a chemical component, functional component, on the devices. We would like today to be able to modify this chemical layer only on the silicon nanowires and not around the silicon nanowires. Everybody today, including myself, prepare devices on silicon oxide surfaces. So actually we get chemical modification on all the surface. One of the projects that we are taking today is actually prepare those devices on surfaces different than silicon oxide or glass, plastic surfaces, and being able to do chemical modification only on the silicon nanowire. And the most important step here, that is one of the biggest challenges, is to get the most homogeneous, but at the same time the thinnest binding layer during this step. The thickness of the step is critical because we are sensing charges, and the debye length in physiological pH, or when we have salts, we have a debye length. Actually, debye length is the minimum distance that two charges can get away one from the other that still will fill each other. So if the thickness of the film is too big, then we won't get any sensing. So we need a very thin film. We need to optimize the chemistry, and that's what we did. So the next step, we can bind an antibody. An antibody is a molecule, or it's a biological element that can bind specifically an antigen. So this is the second step. Then we should protect or chemically bind or protect all those aldehyde groups, free aldehyde groups. So we won't get during the sensing step binding of biomolecules into the free aldehyde groups. And the last step is the sensing. So actually the most critical step, like I said, is to create an homogeneous layer of silane molecules or silane derivative on the surface. So actually we could follow by using AFM, in situ AFM during the modification of silane. we could follow the thickness of the layer of the Silane with time. And at the same time, we could follow the sensitivity of the device for each one of the thicknesses. And we see clearly that as the thickness of the Silane layer, or the sensing layer eventually, is thicker and thicker and thicker, then we get a dramatic decrease of the sensitivity of the devices. So we would like to be in this region where the thickness of the Silane is between 1 to 2 nanometers, and we get high signals.
What is the thickness of one layer? One layer of Silane.
One layer of Silane depends on the Silane. If you use Aptes, okay, in this case, this is approximately half a nanometer. Half a nanometer, more or less. So it will be like Silane, silicon, then you have CH2, 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 aldehyde. Approximately half a nanometer, a little less. Those molecules are not rigid, so also the AFM measurement won't give you a real value. But it gives you an approximate value, more or less, of that the reaction is going on, and you get a thicker and thicker film with time. So then the next step is to be able actually to characterize or count the surface density of or the number of antibody molecules that we have in a single nanowire. That's a challenge because that's very difficult to see a nanowire. That's very difficult to see antibody molecules by any technique. So we need to find a way to label the antibody molecules so we will be able later on to count them. So the way was that I prepared a conjugate molecule that approximately have a single nanodot per antibody. So actually, after binding the antibodies to the nanowire, we can actually go to TEM, transmission electron microscopy, and count nanoparticle by nanoparticle, and approximately know the number of antibody molecules on the surface of the sensing device. After we have the devices and we know what is the range and what are the best or close to the best conditions for the best signals, we can do sensing experiments. In this case, we have two devices that were measured simultaneously. The first device was modified with an antibody that is specific to a molecule, to a protein that is called PSA, prostate-specific antigen. That's a known cancer biomarker for the detection of prostate cancer. The other device was not modified with any antibody, was only blocked with ethanolamine. So upon injection of the PSA molecule at different concentrations, we can see clearly changes of the conductance. In this case, the conductance increases for a p-type silicon nanowire devices, as expected because the pi, or the charge at pH 7 of the PSA, is 6. It means that at pH 7, this molecule is negatively charged. It brings a negative charge to a p-type device, so it will cause an increase in the conductance. And different concentrations of the PSA leads to different signal sizes. For the second device that was measured simultaneously, we don't see any signal, and it means that the sensing step is as specific as we expected and selective. The next step is to modify the same chip with p-type and n-type devices, silicon nanowires, and we see that the sensing of the same protein for p-type devices leads to an increase in the conductance. For n-type devices, we see that for each injection to a decrease in the conductance as expected. So we have an internal control that we know that those signals are coming actually from the molecule that we know that we want to sense. And the next step actually to test that those devices can be used in physiological and blood samples, clinical samples, we took blood samples from different species, human, donkey, and we could clearly see that we can detect very low concentrations, extremely low concentrations of this cancer biomarker, PSA, in real samples. And I want to emphasize that real samples, the concentrations that we sense in this case, we can see from the concentration dependence plot, are down to the femtomolar level. In a blood sample, we have concentrations of proteins, total protein concentration of around 50 milligram per milliliter. So we have around 10 orders of magnitude more proteins in the blood sample than the concentration of the protein that we can actually sense. So it means that our devices are not just very sensitive, are also extremely selective. And the selectivity is actually a factor of the antibody. The antibody actually, he dictates the selectivity of the devices. The next step is to put more devices on a single surface, and we modified, in this case, a number of devices on the single surface with different antibodies for 3 different cancer biomarkers: PSA, CA, cancer-embryonic antigen for detection of colon cancer, and Muci, that is a known cancer biomarker for the detection of breast cancer. So we can clearly see that when we inject on this surface PSA, only the devices that were modified with the antibody against it will give a signal, and the other devices do not give any signal. And the same happens for the other 2 devices when we inject sequentially each one of the biomarkers. The next step is to actually make this multiplexing system even larger and broader. So we want to find a chemical technique, a mechanical technique, a robotic technique that actually can drop very small drops of biological molecules. So in each drop, we can modify a number of devices, a single device or a subgroup of devices with different antibodies, each one with a different antibody. So the same surface will be able to sense tens or hundreds of proteins in a single shot. So actually, this technique of liquid dispensing is known. People prepared in the past, already many years, DNA microarrays by using the same printing techniques. You have a small tip with a small liquid meniscus. It comes close to the surface, and by touching the surface, you get a small drop on the surface, depending on the size of the tip. So we can modify, by using this tip, the surface with many, many drops of different biological molecules. So then we can simultaneously detect different proteins in a single shot. We can see each color is a subgroup of devices that was modified with a different antibody. So we can see that when we inject PSA, those threaded nanowires, PSA, to the system, only the nanowires that were modified with a PSA drop gives a signal. All the other nanowires do not give any signal. And the same happens for all the other proteins in the system. We see the concentration dependence also, so we get concentration dependence for all the cases down to the femtomolar level. And the chemistry that we can do on the surfaces is very broad. In this case, I took this system because it is a chemical challenge. Actually, the detection of toxins. Toxins are actually proteins from bacteria that binds on the surface of cells and cause lysis of cells and destroy the cell, kill the cell. So actually, those toxins bind to receptors on the cells, on living cells, and those receptors are embedded, are stuck into the membrane. So in this case, the receptors are mainly what we call glycolipids, gangliosides. That's a molecule that is very hydrophobic, long hydrophobic change, and at the end, they have a sugar group. The sugar group, this part is inserted into the membrane. The sugar group is showing out the membrane. So actually, we need to mimic that system in order to be able to sense the toxins. So the idea was actually to bind or to create a monolayer, a carpet or very hydrophobic carpet on the silicon nanowire, and then to mix it with the ganglioside molecule. The ganglioside molecule inserts into this carpet exactly as it does for biological membranes. That's actually like a membrane mimic. So then, at the last step, we can sense the binding of the toxins to this monolayer. We can see here 8 devices sensing different toxins simultaneously. Cholera toxin, botulinum toxin, ricin. I won't show that so much. The only thing is that many people ask us all the time questions regarding the kinetics of binding. We were able to show that we can follow, as people do for SPR, surface plasmon resonance at QCM, we can follow and we can measure kinetics. And the main importance of our system is that we can follow kinetics because of the ultra high sensitivity of our systems. We can follow kinetics even when the concentration of the biological molecules is very, very, very low. So that has a great importance in many areas, like pharmacological systems, diagnostic systems. When you want to follow the mutation of cells and the release of small concentration of molecules from cells in real time. So we can follow kinetics. And we not only can follow kinetics, we can also do multiplex kinetics. We can follow kinetics of the same protein that binds to a large number of different receptors for the same protein on the surface. Or we can use the same receptor and follow the kinetics of the binding of a large number of proteins to the same receptor at the same time. Other systems are, for example, following DNA extension reactions. We can follow on silicon nanowires the polymerization reactions of many proteins. And we can sense also in this system single viruses. The devices are so sensitive that the binding of a single influenza virus, as we will see now, in this case, will cause a deflection or a change in the conductance of the devices. We can see that the system is stochastic, the sensing is stochastic. Each time that the virus binds and unbinds from a single nanowire, we get a spike that it depends on the time the virus sits on the nanowire until it leaves again. So each nanowire should give spikes at different times. It cannot be that two nanowires will give at the same time the spikes, because a single nanowire cannot bind to two systems. A single virus cannot bind to two systems simultaneously. So we see here two nanowires binding influenza viruses. Increasing the concentration of the viruses leads to a larger number of spikes. We can actually know the charge of each viral particle by changing the pH. This is very difficult today to know the charge of cells, single cells, or single viral particles. People do a very complicated technique that is called isoelectric focusing. We can do that very easily by our technique, changing the pH. When we get that the signals from the same virus turns to be from negative to positive, we know that at this point, around this point, the nanoparticle, the particle is zero charge. So, and we can also control the time that the virus particle will stay on the surface by changing the surface density of antibodies against the virus. Actually, the virus is like a cell, it's like a small system that contains a large number of proteins, of the same protein on the surface. So by making the surface density of antibodies to this protein larger, then the virus can bind to many points on the silicon nanowire, so it will stay longer there. So we can see here other systems where high antibody coverage will lead to almost non-reversible viral binding. So we can, at the same time, sense influenza and AIDS, HIV, on a single surface with a sensing limit of a single virus. And that's all.
We have time for questions.
Nice. Okay. I enjoyed your lecture. I have two questions. First, you showed a membrane in some of your previous slides, a membrane, and the detected molecule was outside the membrane.
Yes.
Was that very far from the Helmholtz layer? Was not screened by the protecting solution?
The pictures always look bigger than the reality. Actually, the Silane is a C. It can be a C12 or C18. So actually, if you see how much is C18, it is like C18. It is like how many nanometers? It will be like 0.2 per 0.2 per each one, 0.2 by 18. So no, the membrane is there. But actually, the Helmholtz layer, if you actually you start to count the screening of the device length in this case, because the membrane is very hydrophobic, actually there is no water inside the layer, hydrophobic layer. So actually you don't count the layer, you don't reduce from this the thickness of the layer from the divide. In the other case, where you use hydrophilic silans that can really get swelled with water molecules, then it is very important to actually see the thickness. That's actually a very good question, because one of the things that we want to work in our group is, first of all, and we are working towards that, is actually, first of all, not to use full antibody molecules, but actually genetically and artificially create new antibody molecules that only contain the binding site of the antibody and not the whole antibody, not all the nonsense parts of antibodies. So it will decrease actually the thickness of the sensing layer. On the other side, to create sensing layers as hydrophobic as we can. So it will lead to much more sensitive devices.
In principle, in self-assembly monolayer technique, you can control very precisely the number of layers that you draw. And in your graph, you sort of describe a continuous process where you control the thickness of the cell. Why is it so? Why can't you do just
working with monolayers on gold? If you have a technique, for example, using a D-thiol derivative, if you use a D-thiol derivative, alkanthiol derivative, on gold, so you can create one layer of alkanthiol, and on the top of that you can do again self-assembly, second layer of alkanthiol. That's known. Monolayers for silane molecules, silane derivatives, are also known, but when you do that in very controlled environments, completely dry environments, and not for every silane derivative. Already when you have a head group that is not hydrophobic, already the monolayer won't be as good, or won't be actually a monolayer. Actually, for aptes, amino-propyl triethoxysilane, those triethoxy molecules that are good living groups cannot only bind to the surface, but can bind to other silane groups that are floating around. So at the moment that this cross-linking can happen on the surface, then you don't have a layer by layer, but you have like, I don't like to call it layer by layer, you cannot prepare a monolayer. You have like a type of gel structure on the surface that grows and grows and grows, amorphous structure. So you can control the thickness, but you cannot control the structure.
Sure.
When you grow the Silane, you use the 95% ethanol and 5% water. As far as I know, in literature, when you use water, you have a tendency to grow a multilayer. And many times when people want to build monolayer, they use the toluene. Did you try to use the
That question is very good. The only problem here is that all those devices has a memory, a chemical memory thing. Toluene is not such a friendly solvent to work with devices. And silicon nanowires, from the electrical characteristics of the sensing devices that I do, they don't look so well after that, or they are not so the devices are not so good electrically. There are many tricks using silane. You can use toluene, and that's a good technique. It has to be completely dry actually, because it's enough some water molecules or humidity already to make, like you say, the tendency for the multi-layer. So there is a trick. You do pre-incubation of the silane in the ethanol 95%, and you leave it for half an hour, what I call the pre-activation step. In this pre-activation step, you get all the fast kinetic reactions of oligomers, short and long oligomers of silane, and this gel-type structure. So after that, we filter the solution. We get eddy of all the oligomers, polymers, and things that the gel type things that stays there, and we get through the filter only the monomers. So you can try it.
I have two short questions. One, what is the thickness of the silicon nanowire? And the second is the binding of the virus or some protein to the antigen reversible? Can you wash it? Is it
Yes. I'm very sorry that I didn't show that. I didn't just... Sometimes I did that so many times that I just forget that. Yes. So you see here, we have the pointer. Okay. We see here that this is the injection of the sample. So we see a change. So then we inject always buffer. So it decreases back to the original level. So always those peaks are actually not happening without injecting buffer. So all the bindings are reversible. That's a very good question. It depends on the binding, on the K-off of the binding of the antibody, or the strength of the interaction between the antigen and the antibody. Other questions that I don't enter into that is, for example, the thickness of the nanowires. They can be as small as 2 nanometers. They can be as big as 200, 300 nanometers, depending on the diameter of the gold nanoparticle. I want to perform some experiments to see actually what thickness dependence, what is the best thickness in order to get the most sensitive devices. On the other side, that's a question sometimes people ask, sometimes not, about the doping. What is the optimum doping? Do you want a lot of doping? Do you want less doping? So actually, that's a part of the work that we still have to do.
Just one very short question. You have?
If you try to use direction of growth of the nanowire, I'm not sure if I've seen silicon nanowire. So you need to try because I think 2 electrodes to the protein state of nanowire and growth
People are trying to do that today. The directional growth is actually, I would define as the holy grail of the manipulation of nanostructures. We don't need to manipulate them. We can grow them wherever we want. Actually, that's not real so much today to get a good yield of devices. In order to make directional growth, you need a p-taxi. Or you need some signal to the nanowire to tell it you grow to this direction. A p-taxi is actually the signal. So to create a p-taxi wherever you want in 200 devices, actually not in the vertical direction, but in the horizontal direction is not easy.
Thank you very much.